stories from my lab. So yeah, today the goal actually for me is not just sort of promoting our work. Actually, I want to make this helpful for a lot of biologists in our community, and because sometimes I get a lot of questions regarding, should we use this? How do we do drug discovery, or should we make degraders, or should we make inhibitors, things like that. So I just want to also goes to those I go to those study sections for drug discovery. See some weaknesses from the grant. I want to share those experience basically. Yeah, just quick disclosure of some of the work we did. Actually, we have financial interest. So I break this up in 3 parts. Actually, there's a lot of content to go through. I just want to make sure we can go through this. So the first part, I'll talk about some chemical probes and review some of the basic concepts of pharmacokinetics. I think everybody is very familiar a lot of biology part, but understanding of pharmacokinetics and PKPD consideration is very important. Actually, to apply this in vivo also for drug discovery. And also the a lot of compounds, they have off-target effects, and you want to select the specific compounds for your applications. And I'll also briefly go over some of the concepts about small molecule drug discovery, including reversible inhibitors, irreversible inhibitors, and also the protein degraders, mostly focused on PROTACs. That's kind of like the popular thing these days. And if you have a validated biological target, how you start your drug discovery project as a biologist. And briefly mention some of the Houston local resources too. And then I'll just briefly introduce the work we did. The first 2 already published. There's some unpublished work also, but I don't have time to go into details, but I'll be happy to chat more if you're interested. So yeah, the first part is about go over some of the basic concepts. So the thing is, for a drug molecule, once they get into our body, the so-called journey of a drug molecule is basically it's gonna go through the so-called ADME process, right? So first it got absorbed, and then it got distributed into the tissue, and then metabolized, and eventually eliminated from the body. And this is so-called ADME. That's very important because the reason we want to understand the PK parameters is because we want to get optimal therapy. If you don't have drug in your system, in your blood, then it's difficult to achieve efficacy. For example, like the caffeine, the half-life of caffeine is about like 4 hours, right? So that basically can tell you how often you need a cup of coffee. So we'll explain that a little bit. So the one very important concept is called half-life, right? So you may heard about this all the time. And so what is half-life? Half-life, basically the time needed to reduce 50% of drug concentration. For example, in this figure, you can see this is the PK plot, right? So this is for a intravenous injection of a drug, essentially. And so if the initial concentration is 100, once they drop to 50%, to 50, it takes about 5 hours. Then from 50 to 25, it takes another 5 hours, right? So essentially, constant percentage of drug is eliminated per unit time. So that's so-called first order kinetics, first order pharmacokinetics, right? So then generally the rule of thumb is if you go, after like 3 half-lives, essentially you only have about 12.5% of drug left, right? Or let's say about 10%. So generally, we will think at 10% is essentially have nothing left in your system. And so coming back to the caffeine example, if the the half-life of caffeine is 4 hours, so that means after 12 hours, only 10% caffeine is in your system, and then you need to, if you feel sleepy, you need a cup of coffee essentially. But another thing you want to remember is that all these PK parameters, they are actually patient dependent, right? Because some people may have less capability to metabolize the drug, and then the half-life could be longer, right? For example, if the drug is metabolized through the liver, and then that patient has liver dysfunction, then the half-life of that drug may be longer than in that liver dysfunction person than the normal people. So another important thing is that if you see this kind of PK curve, so when you reach the maximum concentration, we call that Cmax. That's very important because that's pretty much the highest concentration you can reach in the blood, right? And I'm gonna give you an example, actually, like sort of common mistakes sometimes you see in grants or in papers. So Tmax basically the time when you reach Cmax. So this is not very important, but it's basically tell you, how long does it take for you to reach the Cmax. But if you give a drug intravenously, generally Tmax is very short, like instantaneously, essentially. If you give it orally, it takes a little bit of time sometimes to get absorbed. If it's a mouse, sometimes take about 30 minutes to 1 hour, but definitely depending on the formulation for sure. And all the blue area is so-called area under the curve, so that actually a parameter to evaluate the drug exposure. Okay. So then what is PKPD, right? So for a lot of, the PK, we just briefly talk about, you can get a curve like that. For a lot of our in vitro experiments, we can get a so-called pharmacodynamic response, or PD, right? So essentially, if you have higher drug concentration, you can see higher response. And if you just basically, multiply these two together, you get this so-called PKPD modeling. So essentially, you know the time, and then you know the response, right? So then eventually, when you are at longer time points, your drug concentration is lower, so then your drug effect, based on this PD curve, will be lower. So that's so-called a PKPD modeling or PKPD relationship. So it seems like pretty obvious to many of you, but sometimes if you see the literature, this is the thing you may always see, like the so-called magic pill, metformin, right? So you can see it's anti-cancer drug, anti-aging, anti-everything. So so the thing is that, for example, just random paper actually I picked. It's a cancer research paper. If you see actually the concentration they use, right, in their in vitro experiment, they use like 2.5 to 20 millimolar. Right? So you want to, it's a millimolar. And then the first question in my mind would be, is that physiological relevant? And so actually there's a very good review recently to talk about the controversy on metformin concentrations. And for in vitro studies, they would say generally, they believe the metformin physiological relevant concentration will be 40 to 70 micromolar. So that means, right, so in the previous study, when they use like 10 millimolar or 20 millimolar, that's not relevant at all, right? No matter what kind of effect you see, that's not physiological relevant. But you actually can see a lot of papers use physiological irrelevant concentration, not just for metformin, but for many other compounds. I think that's definitely a big if you do that in a grant application. So this is basically example to tell you how do you know what the Cmax or maximum concentration you can achieve for metformin. Metformin is an approved drug, so you can quickly actually look it up, do a Google. You just Google metformin Cmax FDA label, and you can come up all this information actually. this is basically listed on the drug label. They're supposed to tell you, the dose, dose frequency, and also the Cmax, right? So for the drug. So I just give you some like very so some examples here, right? So they say the peak plasma level approximately like 0.6 to 1.8 microgram per ml. There's another report, they said the maximum, no matter what kind of dose you give, the maximum concentration Cmax is 4 microgram per ml. So when you see that, so you basically need a molecular weight of metformin and then convert that to concentration. As a rule of thumb, so for me, how do I do the math? So I remember this, I memorize this. If a drug has a molecular weight about 100, if the concentration is 1 microgram per ml, actually that corresponds to 10 micromole, right? So then if you increase the molecular weight, then the concentration will decrease. And if you increase the concentration will also increase proportionally. So if you do that, then you can calculate for metformin, 4 microgram per ml corresponds to about 30 micromolar. So this is consistent with the previous review mentioned, the maximum concentration for metformin is 40 to 70 micromole. So this is basically, if you use other drugs, right, especially FDA-approved drug, you can easily find all this information. If not, you also may want to check whether Cmax has been reported in the literature. Because if you don't use that to do the in vitro experiment, and the whole point of doing in vitro is to predict the in vivo behavior. So there are so many chemical probes out there. So how do you choose the appropriate one essentially? So this is actually a very good website portal. It's called chemicalprobes.org. This one, actually, they have a lot of chemists and biologists. They review these reported chemical probes, and they will actually rank them and give you a score. So I think 4 is the best score. And they also tell you whether this compound is suitable for in vivo applications or not. I think if you're looking for any probes, this would be a good place to check first. So then if you work on kinases, actually there are a lot of resources out there too. This is a website from Harvard Medical School. It's called the Kinome Scan database. So essentially, they list a whole bunch of whole bunch of drug molecules here, and they basically scan the whole kinome, see whether this compound is specific or not. And if your compound of interest is in the database, you can quickly know how many kinases this compound can hit and potential off target. For chemical probes, we would prefer to have the more specific, the better, right? And then that will simplify the biological interpretation. This is actually a very good review. I really like it, written by Black and Workman. It's called Choose and Use Your Chemical Probe Wisely to Explore Cancer Biology. But this is definitely the concept here can be applied to other research too. So they give you a list of do's and don'ts. And so, I mean, they're a part of this chemical probes portal. So they trying to promote their portal, which is actually a great resource. And so they said, check that first, right? And also, they recommend check with chemists, right? Or experienced medicinal chemists to look at the probe you want to use. And do not just rely on vendors for sure, and also do not just follow what other people use, because you can see sometimes the person actually didn't know how specific the compound is, and then he used that, and then other people keep citing that paper, use that probe, and sometimes it can be misused or give you wrong interpretation. So just be very careful.
Wonderful, small, nimble, flexible, trendy, a little edgy, but very.
Hello? Any questions?
So if you do the drug combination, you have to figure out, say you do A and B, you have to figure out both, right? Or do you need to consider their effects separately on,
you're talking about trying to use
combination
chemicals to probe biology, or you're trying to do drug combination?
Drug combination. So my question is, when you consider drug combination, essentially, could you just using the same for individual drug?
Right. I think my take for that is for probes to study biology, the cleaner the better, right? So the more specific the better. But for drugs, sometimes it's not necessary, because sometimes the so-called polypharmacology is a good thing to have, right? So for example, you have a dual inhibitor for both EGFR and HER2, that could be better than just you only hit HER2. So I think if you're considering drug combination, that's a little bit different from selecting probes.
Okay.
Then I will just go to briefly talk about these small molecule drug discovery and go over some of the concepts here. Yeah, this is actually the most popular theory, so-called ligand receptor theory for pharmacodynamics, right? So essentially you have a receptor, and your ligand can bind to a receptor, trigger all this cascade, and then trigger a biological response, right? Either cell death or whatever consequences here. What is actually reversible inhibitor? These are actually the most commonly compound everybody uses. So for example, you have a small molecule, actually they can bind to a receptor, bind to an enzyme through this so-called non-covalent interactions, right? So that will include hydrogen bonding, hydrophobic interactions, or stop rate or things like that. So that's so-called non-covalent interactions. So the thing here is an equilibrium is established between the free ligand and the ligand receptor complex. Okay, so remember here, this is the equilibrium, so it can go both ways. If you have a lot of ligand, it will push the equilibrium to the right. If you have less ligand, and your ligand receptor complex is going to dissociate and shift the equilibrium to the left. So it's always the equilibrium. So that's very important to remember. And so essentially, if you do dialysis, right, and you can remove the small molecules, then you can restore the activity of the receptor completely. And another important thing here is if you have one receptor, at least you need one molecule of the ligand, right, to inhibit that receptor. So this is so-called occupancy based, right? So one receptor need at least one ligand to occupy this. And I will tell you why this is important, and how it's different from degraders in the next couple of slides. So because of this equilibrium or this reversible process, the pharmacokinetics and pharmacodynamics are tightly correlated with each other, right? So once you remove the drug from the system, your pharmacodynamic response is going to decrease too, because you shift the equilibrium to the left. So this is basically the the most commonly the classical PKPD relationship. So then what are the irreversible inhibitors? Right? Irreversible inhibitors, actually ligands will form covalent bonds with receptors through reacting with amino acid side chains. For example, you have a cysteine, lysine, tyrosine, serine, so on and so forth, right? So as long as these side chains have certain reactivity, you can design a covalent inhibitor. So essentially, if you have this irreversible inhibitor, this reaction will only go one way. Okay, So once you form the ligand receptor complex, even you do dialysis, remove all the ligand, it will not dissociate. It's stuck there. So this is important. Another thing is, again, this is also occupancy based, because you have one receptor, you need at least one ligand, right, to occupy that receptor to have the effect. And because even you remove all the ligand from the system, right, so then the PKPD are not coupled. Basically, they decoupled from each other. That means even you remove your drug from the blood, and because your ligand still stuck to the receptor, then you may still maintain the pharmacodynamic effect, right? So then what depends on the PD, the pharmacodynamics? Basically, the protein resynthesis rate actually determines the pharmacodynamics. So essentially, once your receptor has its own certain turnover rate, right? For example, 12 hours, then that means after 12 hours, you have to give the drug again, because those receptors going to be inhibited receptors going to be degraded, and then new receptors going to be generated, and then you need drug to hit them again, essentially. Common examples for irreversible inhibitors. This is pretty interesting. Omeprazole actually is a proton pump inhibitors, right? If you have like, it's a commonly used so-called acid reducer if you don't feel well in your stomach. So you can take this. This is actually one of the early examples of irreversible inhibitor. Actually, I didn't know this until I learned all the details. It's pretty interesting. They will essentially, you have the compound like this, and then this compound actually get activated under acidic condition, form this structure. And this structure, then they will inhibit the proton pump, react with the cysteine residue in the enzyme active site, form a covalent bond here. So essentially, this is how omeprazole works. There's another very famous example for irreversible inhibitors from ibrutinib. This is actually the first approved, FDA-approved irreversible kinase inhibitor. So at the time, everybody, actually nobody really liked irreversible inhibitor, because they would think, they thought if the, this compound non-specifically react with any cysteine in your blood or in cells, then they will stuck there, right? So you may have a lot of off-target effects. But the thing is, it all depends on how reactive the microreceptor or the irreversible group is. If you make it less reactive, then you have to have the molecule actually bind to the binding site. Then the cysteine residue can react with this so-called microreceptor, the double bond here. So this is actually a very popular drug for BTK, protein tyrosine kinase inhibitor. So I think the sales is like in the $1 billion range each year. Okay, so those are the small molecules, the reversible inhibitor, irreversible inhibitor, and I got a lot of questions from friends and also in our SCBA community about these degraders. So we recently started working on these. Actually, they are these are pretty interesting compounds. So let's just take a quick look at the protein degradation pathway. You guys know much better than me. So protein can be degraded mostly through two pathways. One is through the lysosomes, and then the other through this ubiquitin proteasome system or UPS system. So today we'll mostly just focus on the UPS system. So in this pathway, essentially, you have the E1, E2, E3 ligases, essentially, and your protein actually get ubiquitinated, and then the protein will be sent to the proteasome get degraded. Right? So that's basically how the protein get degraded through the endogenous machinery through this UPS system. So as to develop therapeutics, we can actually hijack this UPS system to develop therapeutics. This is actually a pretty good review written by the George Winter's group. So he basically summarized the 4 types of these scenarios for small molecule induced protein degradation. So the first one is so-called a PROTAC, which I would explain in more details. This is essentially a bifunctional molecule. You know, one end of the molecule can bind to the E3 ligase, the other end can bind to your protein interest. So then you bring this protein interest and E3 ligase together, and this protein interest get ubiquitinated, and then the protein got degraded. Right? So that's basically induced by a bifunctional molecule. These are actually a relatively large molecule, just like Mike asked me at the very beginning. The molecular weight of these generally about 1,000. There's another kind of degrader. These are actually very, very good compounds. We call them molecular glue. So essentially, this type of molecule is just like a small molecule. It's not a bifunctional molecule. It's just like a small molecule, with molecular weight about 400 or below 500. But the interesting thing is this molecule can work as a bridge between 2 proteins, between the E3 ligase and the protein interest. If you don't have this small molecule, these 2 proteins actually will not interact with each other. Only when you have this molecular glue, you can glue them together, and then you drive this interest to the E3 ligase, and then get ubiquitinated, get degraded. The most famous example for this is thalidomide, right? So thalidomide actually can induce degradation with transcription factors, AKT1 and 3, and many some other transcription factors. And there's a recently discovered these sulfonamides. Actually, they can also work as a molecular glue to induce degradation, protein degradation. So these are basically kind of like I should say, these are therapeutic, like drug-like molecules, right? So you basically throw the molecule directly into cells, and then will trigger protein degradation. But to develop these molecules that are not easy, right? So it's not it cannot be a general strategy to induce a protein degradation. So for example, if you have a protein, you wanna use this protein degradation strategy to study cell biology. So what can be a general strategy to use? So there are 2 ways. One is called a DTAG strategy. So in this strategy, you have to create a fusion of your protein interest with a DTAG domain. Okay, so you make a fusion. You can do CRISPR knock-in, or you just overexpress, depend on how important the protein concentration is to your biological effect, right? Preferably, you want to do the knock-in. And then you can add in this case, bifunctional molecule. And so this molecule, one end can bind to the DTAG domain, the other end bind to E3 ligase. Then your protein interest get ubiquitinated and then get degraded. Or with similar concept, you use this auxin-induced degradation system. You have to have this auxin domain, right? Also make a fusion protein. Then you add this small molecule, and then you can trigger the degradation. The thing here is for both of these 2 strategies here, you have to engineer your cells, basically make them as a model system to look at this. And you may ask, hey, I routinely do siRNA knockdown or CRISPR knockout, right? So why I need to look at this protein degradation? why I want to use this protein degradation strategy. So the thing is, for knockout, you basically permanently knock your gene out, right? So then you may have a secondary effect. Cells may adapt to the knockout and then ramp up other machinery. So then the biological readout you see may not be the primary effect. But here with this protein degradation strategy, the protein can be degraded within hours. So you can, you will be able to look at the primary effect only. So that's basically the advantage. So what are the PROTACs? PROTACs, actually the full name is called the proteolysis targeting chimera, or PROTAC here. This concept was developed by Craig Cruz in the like about 20 years ago. So essentially, the idea here is that you can use a bifunctional small molecule, and the one end you can bind to a target protein, the other end bind to E3 ligase. You bring them together, like here you form the so-called ternary complex, right? So 3 molecule complex, ternary complex. So although the cartoon is drawn like this, but the protein interaction between E3 ligase and the target protein is also very important. That means for a specific target protein, you have to find a proper E3 ligase to degrade it. And this process is highly empirical. There's no rational design so far, right? So it's all trial and error. Once you form this ternary complex, and your target protein can be ubiquitinated and then get degraded through the proteasome. And then your small molecules will be released from the ternary complex, right? So then it can go back as a catalyst to degrade another target protein. So remember in the previous slides for reversible and irreversible inhibitors, I always emphasize those are occupancy based. Means with one protein you need at least one molecule, right, to react or bind to your target protein. But in this degrader case, it's not occupancy based. You can have only basically theoretically only have a few molecules, say 2 molecules, and then you'll be able to degrade 100 molecules of your target protein, because this is a catalytic process, right? So that's the key difference between these degraders and those inhibitors. So what are the pros and cons of PROTACs versus inhibitors? PROTACs get like so popular these days. Again, this first is the occupancy, right? Stoichiometry. So for traditional inhibitors, you need 1-to-1 at least, but for this PROTACs is catalytic, so you don't need to reach that stoichiometry. Drug exposure. Again, for reversible inhibitors, you need the constant drug exposure, because if you remove the drug, then you lose the pharmacodynamic effect. But for PROTACs, because it triggers protein degradation and the drug exposure is not constant drug exposure is not required, and basically the pharmacodynamics determined by the protein resistance rate, right? Just kind of like irreversible inhibitors. And in terms of function, right? So inhibitors basically can only inhibit one domain of the protein, but then for degraders, you can just get rid of the whole thing, which sometimes could be beneficial. And specificity, right? So inhibitors, the specificity comes from the small molecule design, but for PROTACs, not only the small molecule design, but also the E3 ligase and the protein interaction is also important. So that's a secondary, basically, check, I would say, right, for the specificity. And then in terms of targets, right? So for inhibitors, it's mostly enzymes or receptors, but for degraders, potentially you can, as long as you can find a binder, and it doesn't have to be very strong. Sometimes micromolar binder is sufficient to trigger efficient degradation. Right. So these are the good thing about PROTACs. But then what's the downside, right? So the downside, the main downside is the molecular weight, and the molecular weight generally is very large. Originally, people didn't believe those are drug-like molecules. And so that's why the cell permeability is a common issue for these PROTACs. And so then for the molecular design, it's still very empirical, just mostly trial and error. So, if you tell me, hey, Jin, make a PROTAC of this protein, I will say, there's no guarantee, right? So we can only try it until we know. And so because of large molecular weight and the ADME property of the PROTACs generally very poor, and the linker sometimes can be easily metabolized. You want to be very careful about that. And also the bioavailability of PROTACs generally is low. But although there are successful examples out there showing PROTACs can be orally available, but that requires a lot of medicinal chemistry optimization. So, then do you really want to jump into the hype of PROTACs, right? So it's kind of interesting. A couple months ago, I went to like NCI study section, and I'm one of the few chemists in there. And when they see PROTACs, everybody open their eyes. Oh, say PROTACs. Oh, that's fantastic. Highly innovative. But seriously, you have to look at this. Whether it's they use it in the right way, right? So the thing is that number one, you want to be able to see different phenotypes between a active site mutant, basically that's to mimic inhibitors, right? And RNAi knockdown or CRISPR knockout, that's to mimic degraders. You want to see difference between an inhibitor and degrader. Otherwise, there's no point of making a degrader, which is takes a lot of extra effort, and then it's highly empirical. Sometimes failure rate is very high. So you rather just go with inhibitor. For example, like a kinase, right? A lot of the PROTACs actually targeting kinases. You want to be able to see there's also non-kinase or scaffolding functions for that kinase to provide survival benefits, right, for cancer cells, for example. Then it will make more sense if you develop a kinase degrader. You expect that will work better than the inhibitor, right? So again, like I said, develop these degraders, a hit and miss, so it requires a lot of labor-intense screening. And due to the poor permeability of PROTAC, their efficacy is highly dependent on the PROTAC recycling feature. This is actually very important point. This is also related to the third story I'm gonna tell you. Generally for PROTAC, because of poor permeability, the drug concentration in cells is pretty low. Essentially, you cannot, achieve the same concentration as your protein target. So then you have to heavily rely on the, the recycling feature. Then, you may ask the question, if I try to degrade a kinase, how much degradation is enough, right? Is it like 80% degradation is enough, or you have to achieve 100% degradation, which is very difficult, right? So for example, if you achieve 80% degradation, then the remaining 20% of kinase may, because it's an enzyme, right? It may be enough to just do its job, and so then you may not be able to see any phenotypes. So that's basically the dilemma of this PROTACs, right? So if you really want to achieve very high efficacy, you want to be able to achieve very high percentage of degradation, which is kind of challenging. So we actually serendipitously discovered a way to develop a so-called dual inhibitor and degrader. Essentially, we find a way to enhance the permeability of PROTACs, which is one of the main issues in this field. So I'll share that story later. So here's a quick summary of these. I don't want to go over this again, but essentially, the difference between these reversible inhibitor, irreversible PROTACs, and molecular glues, and the difference. Number one is occupancy, right? And then the PKPD, whether they are decoupled or not. And for inhibitors, they mostly only go after one domain, but for degraders, you can get rid of the whole protein. For specificity, for inhibitors, they mostly rely on just a small molecule design. But for degraders, you will not only on the molecular small molecule design, also the protein-protein interactions, right? Which means the ester ligase and your target protein interaction. That's also very important. So in terms of targets, so inhibitors will be generally regular targets you always use, but for degraders, you may see you may expand the targeted proteome. And inhibitors, you can rationally design them. You can do screening, but for PROTACs, it's highly empirical. And for molecular glues, actually, I would say God knows how to come up those. And so far, these molecular glues reported so far, they all discovered, serendipitously. So like thalidomide, right? So initially, people didn't know why it works, and through a lot of elegant work, they discovered, oh, it works as a molecular glue. So far, this is a highly challenging area to develop or to design, rationally design molecular glue. So right now, it's all come up by chance. So that's why we mostly focus on PROTACs. At least you have some sort of idea how to design that. So if you have, oh, we are almost running out of time. So yeah, if you have a validated target, if you know the structure, if there's a X-ray structure out there, I would suggest that docking is easy and quick, right? And then if you don't have structure, then you can do screening, and you can do high-throughput screening. And Texas A&M has a separate core, or you can work with NCI. They provide service for high-throughput screening, but I think the line is pretty long for that. Or actually, Baylor has a Center for Drug Discovery. They have a technology called a DNA encoded library. You just provide them the protein, they can screen billions of compounds actually. But doing screening or doing docking is all, I would say, the starting point. Actually, the most time-consuming part is the final chemistry optimization. You have to find a chemist partner to work on this, which actually is I would say, highly committed process. So I'll just quickly show like 3 stories, more like, just give you very basic ideas of what we did in the lab. So the first one is this reversible reaction-based glutathione probes, as some of you guys actually use this probe. So what we have here is this is a coumarin, and then we have this so-called microreceptor. It looks familiar, kind of like the one in the ibrutinib, but we have actual CN group here, the cyano group here. That makes this microreceptor less reactive. And then the the so-called KD, the dissociation equilibrium constant, in the millimole range when they react with GSH. Right? So yeah, because we don't have a lot of time, I just want to skip all the details. So you can use these probes. They can be applied directly in live cells. You will be able to image both the blue channel and green channel. Then you use the blue divided by green, and you get this so-called ratiometric map. And based on, you can also do standard curve. You can calculate the glutathione concentration in cells. This is basically one of the experiments we did in HeLa cells. You can see initially it started at 5 millimole concentration with H2O2 treatment, just trigger some artificial oxidative stress. The GSH dropped to about 4 millimole, and then later we'll come back to like 5 millimole. Also, the time response is very fast. It's in the minute range. And we also compare our results with the cell lysate data, and we can get very nice correlation. So yeah, this is probably some of you guys know the story, right? So this paper, we started the whole project about 2013. We fought very hard with the a lot of the reviewers, because this is something I basically came up like randomly, and I know nobody in the field, right? So no name recognition at all. So, but eventually it got published. And at the same day, my daughter was born. So that's why I can remember when this paper got published. So right now, there are more than 100 labs using our probe, and this is now the updated map, but yeah, this number is correct. And you can actually get these probes from a reagent sharing website called CarrierFast. I mean, for people in Houston, just email me, I can just give it to you. And yeah, basically we do that is mostly to avoid MTA process. We also founded this so-called chemical biology probes, and we got a SBIR phase 1 grant recently to further commercialize this. Okay, the second story is just very quick summary of our collaboration with Bert O'Malley's group at Baylor on this drug discovery effort for steroid receptor coactivator 3. So this protein is actually just one of the proteins. It's very floppy. It has a intrinsic disorder domain, so there's no crystal structures out there. O'Malley's group basically did a high-throughput screening with NCI, and they screened about more than 400,000 compounds. This is basically the screen, right? So then they do this high cutoff. Each dot is basically one compound. So once they are done, they brought the data to me and then asked me to see which one to pick, right? So I mean, doing screen is the first step. Then the follow-up is very important. Sometimes you need to have some sort of chemistry sense to look at that. So long story short, ISI-1 is a compound came out of this screen, and the potency of this compound actually in this triple negative breast cancer cells, this is actually 231, lung metastasis 3, whatever. So it has about 200 nanomole IC50, right? So if you, then we did medicinal chemistry optimization, we can reduce the IC50 by at least 10-fold to like 16 nanomole. And we also tested this ISI-2, it can inhibit the SRC3 involve the transcription. This is the P-bind transcription assay. Interestingly, actually, we can induce the SRC3 protein degradation. It's kind of like molecular glue, probably. When we started doing this molecular glue concept wasn't that popular yet. And also, we validated the in vivo efficacy. This is basically some triple negative breast cancer model for 68, and we can inhibit the tumor growth, and we can also inhibit SRC3 in the tumor, as shown here, and also the proliferation. So that's all published like a long time ago. So essentially for this project, right, so O'Malley's group start from high throughput screening, and then find this ISI-1, and so then we can come in to look at this and increase the potency and develop ISI-2. But ISI-2 actually has a relatively poor plasma half-life. The half-life is about 1 hour. So that's why we did like twice injection per day when they did this experiment. So that was pretty intense. And then we from there, it's a poor PK, we modified this. We came up with this so-called ISI-12. Unfortunately, we got some money to work on this. And then we found, we were excited because ISI-12 has a plasma half-life like over 10 hours to 10 to 12 hours. That looks good. But then later we found, ISI-12 has some sort of cardiotoxicity, inhibits the hERG channel. That kind of disappointed us. Then we, went back and developed this so-called ISI-10. And at that moment, we started to talk with outside investors. We formed a company and got more money to further accelerate this process, right? So then when we have ISI-10, we later found, although it solved this hERG activity problem, we lose the PK property. So basically, the PK of ISI-10 is worse than ISI-12. Then we went back to the, to look at where the metabolism happened. We fixed the metabolism, so-called soft spot, and then we came up with so-called ISI-188. It has no hERG inhibition, even better PK property than ISI-12, and so we are actually going to test this in vivo models. We also tested these compounds in the so-called secondary pharmacology assays or syrup assays. These are actually kind of relatively expensive assays, about $5,000 per compound. They will screen about like over 80 like targets, commonly confer drug toxicity. And then if you don't hit those targets, that's considered pretty good. So for us, ISI-10 only hit like 2 targets at high micromole concentration. So that was good. Then very quickly, I'll talk about this PROTAC story. I will just tell you the conclusion here. So here, the target we work on, this is the protein tyrosine kinase. This kinase is very interesting. Actually has a cysteine in the active site or close to the ATP binding pocket, cysteine 481. So that's why you can develop these irreversible covalent inhibitors. Right, so these are approved irreversible covalent inhibitors. The number one is called ibrutinib, I just mentioned previously. For us, actually, we just basically try to convert these BTK binders and conjugate them with the E3 ligase, like this thalidomide bind to cereblon, and then we compare different chemistry here, right? So one, we just get rid of the irreversible reacting group. We make a single bond here that becomes a reversible non-covalent binder. Or we can use the irreversible covalent, just like ibrutinib, right? And because of our work in the GSH part, we developed this so-called reversible covalent chemistry. So the idea here is, when this compound in cells, the binding to thiol is very weak. Only when you have the BTK pocket, the BTK can bind to this part of the molecule, then the cysteine can react with this microreceptor, form a covalent bond. So originally we just want to compare the 3 chemistry, but we found something very interesting, which was unexpected. Yeah, I'll just skip that. So if you compare the protein degradation, they actually the reversible covalent, we call the RC compound, works the best. If you just look at the purple bar, right, it works better than the other 2. So then the question is why it works the best, right? So we basically use this in-cell target engagement assay here, so-called a nanobread assay. And basically the way it works is you can fuse your target interest with a nanoloop. Then you can conjugate a dye to a small molecule, right? So when the small molecule binds, the nanoloop can transfer its energy to the dye and then emit this red emission. But if you have other small molecule compete off this probe, then you will start to see the blue emission, which is due to the nanoloop. You don't have the energy transfer anymore. Right? So then we basically did that. We found actually the RC, reversible covalent PROTAC, give you much higher targeting engagement at lower drug concentration. That means you have more drug in the cell. And we did not only on the cereblon thalidomide side, we also did on the BTK side. We saw the same conclusion. Basically, we have a much better target engagement for this reversible covalent inhibitor compared to the reversible non-covalent and irreversible. And we also compare with many other BTK degraders reported in the literature. We found our compounds is better or similar potency to other compounds. The blue curve is our compound. And also we measure the pharmacokinetics of this compound. RC1 has about 3 to 4 hours plasma half-life, which is okay. now great, we can also degrade BDK in mice, basically in the in B cells. That's just a pharmacodynamic readout. And can this be generalized, right? So then we apply this reversible covalent strategy to another PROTAC, FLT3, and we can see the same phenomenon. Actually, the reversible covalent give you a much better target engagement. Actually, just as good as the small molecule inhibitor, kinase inhibitor. So, the take-home message here is, like I mentioned, for a lot of these, say, the kinase degraders, they have very low concentration in cells, so heavily rely on the degradation, right? So because if you cannot degrade the kinase by 100%, the residual kinase may just work well to play its biological function. The selling point for our system is it's not only a degrader, it also works as an inhibitor, right? So you can degrade, perform degradation of the kinase target. At the same time, for the remaining undegraded kinase, they will be inhibited because we have very high drug concentration in the cell, as high as the small molecule kinase inhibitor. So yeah, I would think this is a new direction for PROTACs. So with that, I just want to actually thank the people who did the work, and also a lot of wonderful collaborators in the medical center. Yeah, just don't want to mention all the names here. And not only this, we also have some other rational drug design projects, which is relatively early, but we got some very promising preliminary data, including collaboration with Hui on the Alzheimer targets. Thank you so much for your attention. Any questions?
So, Jim, for your the GSH probe, I think it's great. There are so many people using them. But are you planning to make a newer version, like for different color, for example?
Yeah, yes, I know, I know. Yeah, we have been trying hard to make different colors. There's a paper reported from a group in China. They came up a red GSH probe. Then I asked a student to reproduce it, and she couldn't get it synthesized. So I think we wanna definitely red color will be better, and probably they can also multiplex with other dyes, right? So that's definitely a direction I'll be interested in. I think right now it's just not very good ideas to address that problem.
Also on the GHS indicator, is it just for TSA? I guess it's also it works for all the reactive oxygen species.
No. So so because of so actually it works with all the thiols, and we call that the GSH probe. It's mostly rely on the high abundance of GSH. If you have, say, very super high concentration of cysteine, it's going to react with that probe too. But generally for cysteine concentration is relatively low. It's about 100 micromole, and GSH is like over 1 millimole. So that's basically our where the selectivity coming from.
So basically, you're just looking at the usage of the GSH in the cell, and for whatever reason, right?
Yeah.
You were using your hydrogen peroxide, to really show the effect, but in reality, cells generate RS under different conditions. Those are the. Now, how does your probe differ from the so the popular sort of the ROS probes out there? There's a lot of different kind of probes, right?
Yeah. So, well, then depends on what you want to probe. If you want to probe hydrogen peroxide, yes, you may want to use hydrogen peroxide probe. So I would say maybe a fair comparison will be how this probe is different from other GSH probes out there. For example, the most common one used is called the monochlorobimine, and that's based on irreversible reaction. So it's basically, you can only, that actually the activity relies on the glutathione S transferase. So essentially that's a GST activity probe. It's not a true GSH probe. And there's another one called a CMFDA. That's actually, that compound is mistakenly used as a GSH probe. It does not reflect the GSH concentration. So actually we are writing a paper to sort of, point that out to the redox biology community. Yeah.
I just have a small question about the small molecule for protein degradation. What's your comment on, in general, the toxicity as well as stability for, I don't know which type I'm asking, but in general, maybe. You
mean the toxicity of degraders?
Yeah.
In general? Well, I think the toxicity is always associated with, depends on, number one, whether your target has, on-target toxicity or not, right? So that depends on, I would say, validation of biology. That's not my work. Then the off-target toxicity, so that can be, eliminated or reduced by better molecular design. So I would say that's very case by case. Then there are a lot of assays, in vitro, in vivo assays to examine the toxicity of these compounds. For example, for IND filing. So those are pretty mature pipelines there. So you just need to plug in your compound into those pipelines and need the money and do it, basically.
How about stability? Normally it lasts for days, hours. What's the?
Oh, yeah. So the stability, again, depends on the molecule, right? So like I said, the linker part is kind of prone to degradation, to metabolism. Actually, we found originally a lot of people use a PEG linker. Actually, PEG linker is a poor choice. It can be easily metabolized. So that's why in our own study, we try to avoid the PEG linker. We make it more like carbon-like. And so there are also studies in the literature. They say they treat the, they use the BTK inhibitors, and then they can see like, I think the pharmacodynamics effect in a week. I don't know. We never reproduce those. We never actually try to reproduce, but I think it all depends on the protein resynthesis rate, right? So if your protein turnover rate is very slow, say, turnover rate is like a week. Yeah, so once it gets degraded, it can last for a week. So it all depends on the protein resynthesis rate.
All right, thanks.
Okay, any other questions?
Gene, in the early part, when you're talking about PD, you're saying that if the compound reactive is a cysteine group, right?
Yeah.
It will be irreversible.
Yeah.
We study ion channel. There's one particular channel called TRPA1. And TRPA1 has a lot of cysteine groups, and it's known that you have quite a number of compounds activate this channel by covalently binding to the cysteine residues. And so they're all considered irreversible, but in our practice, the effect is actually reversible because, you don't really record channel for, hours. You record it for, a few minutes. And you add, in this case, mustard oil. That's a very reactive compound to cysteine residue. And then you wash it for, maybe 2 or 3 minutes, the effect is actually gone. So what's your explanation? If it actually form a covalent bonding, it actually becomes.
So first I want to see the structure of your compounds, right? So maybe yeah, we can do that offline. So all these so-called irreversible inhibitor, technically, even the ibrutinib, it's a microreceptor. So those actually, they call that irreversible because the dissociation rate is very slow. So essentially, it's irreversible. But on this thermodynamic term, is still a reversible inhibitor. It's just like the reverse process is so slow. So then it just depends on how I think it all depends on the reaction probably, and I don't know. I think for your, it's a biochemical assay, right? There's no turnover of the TRP1 channel.
Well, I think at the time is too short for protein turnover.
Okay, yeah.
Okay. So it has to be the same channel,
Right.
Because it can be reactivated. So it's not just the inactivation of the channel itself. So so I'm always curious into what extent and that the so-called irreversible reaction has been reversed. And is it possible maybe these are actually reversed by the glutathione in the cell, or is it?
I mean, for this reaction, probably no, because again, this irreversible reaction from here to here, if you can see my cursor, so that's a unimolecular reaction, right? So it only depends on this molecule, has nothing to do with glutathione. Glutathione just to trap this and then make sure it doesn't go back again.
Okay, so once the reaction happens, you can't really reverse it with the glutathione.
Right. I mean, or say glutathione doesn't do anything. It could be reversed, but just reversed by itself, right? Glutathione cannot accelerate that reversible reaction. So, another possibility is, for example, like this ibrutinib. Actually, the reaction between the thiol to the microreceptor is relatively slow. Slow means may take like hours or say 1 hour. So then potentially for your compound, it's possible, although those compounds look like irreversible inhibitor, but because the time frame is so short, they only bind to the receptor, non-covalently. The irreversible reaction has not happened yet. So then it can be washed out that way. So I think that's potential possibility. So you probably want to do a, this kind of physical organic chemistry to figure out the reaction rate and reversible reaction rate. We can help you with that. We love those.
Okay.
Yeah, that's what I did as a graduate student.
Oh, okay.
Yeah.
Jing, very nice talk. This is Yuben. I had a general question about the PROTAC approach.
Sure.
So, so you have designed to targeting the BTK, for example. And then what about the BTK binder? Binding partners. Every protein has its protein interaction network. Will the whole network be degraded, or only the target will be degraded?
Oh,
so
you mean like
injecting the whole complex into proteasome? Machinery, right?
That's a good question. So you mean like right? So if you are when you are degrading BTK, what about the protein which binds to BTK, right? Are they gonna also drag to the
right? So otherwise, it has a lot of side effects actually.
Yeah, that's a
thinking
good question. We have done a proteome analysis. I deleted that slide, but we actually can see the degradation was pretty specific. We only degrade for the RC1. We degrade another kinase, and that's it. And we didn't see other BTK binder get degraded essentially. So yeah, so you can profile that using just a proteome analysis.
Okay.
Yeah. Yeah, I deleted that slide, unfortunately. Okay. Good. All right. No questions?
Well, let's thank again Jean for the wonderful presentation. Yeah. Thank you.
Yeah, thank you. Thanks everyone.
Next week, Jin Song Liu is going to give a talk on his favorite topic.
Yeah.